De volta, de volta com a nossa programação para sequenciarmos as nossas atividades. Teremos agora um Keynote. Estamos de volta agora sim com a nossa programação para sequenciarmos as nossas atividades. E teremos agora um Keynote. Esse que é o Keynote de número 4, o último também das nossas atividades. O tema desse Keynote será biomateriais sustentáveis para uma biofabricação verde. E nós vamos acompanhar a partir de agora com o Giovanni Vosi. Ele vai trazer todos esses detalhes para a gente sobre esse tema. Giovanni formou-se em engenharia eletrônica em 1998 pela Universidade de Pisa. Obteve o PhD em bioengenharia no Politécnico de Milão em 2002 e atualmente em 2002, né? E atualmente ele é professor titular em bioengenharia. Foi um dos fundadores da Sociedade Internacional de Fabricação, sendo o seu atual tesoureiro. Ele é membro do Conselho de Administração do Grupo Nacional e de Bioengenharia, e seus principais interesses de pesquisa são biomateriais, engenharia de tecidos, medicina regenerativa, biofabricação e modelagem de células in sílico. Vamos, a partir de agora e nos próximos 45 minutos, vocês ficam com o Keynote com o Giovanni. Seja bem-vindo, Giovanni.
I'm Giovanni Vazzi, and I'm professor of bioengineering at the University of Pisa. And in this talk, I try to show you how we can develop novel biomaterials using or recycling waste product. The typical approach of tissue engineering foresee that in order to develop a 3D functional structure that is able to mimic the principal features of a natural tissue can be obtained combining three different stimuli: topological, biochemical, and mechanical. Topological, it's important because its tissue, each organ in our body is able to be functional if the structure is able to tolerate the forces and also the stimuli that are inducing by internal and external forces in our body. Biochemical stimuli are important because it's important to develop a scaffold made of biomaterials that is able to promote the cell activities, but also that is able to activate the cell functions such as adhesion and proliferation and differentiation. And also the mechanical stimuli, it's important because each tissue has a well-defined elastic modulus that allow to the mechanosensors that are present on the cells to activate all the metabolic pathways that are present inside them. Typically, to produce this scaffold or this 3D structure, it's important to combine biofabrication technologies and biomaterial for novel formulation in order that the combination between these two approach or to these two techniques can be able to produce, as you can see from the slide, a well-defined structure where the cells can grow and proliferate. The typical biofabrication techniques that is used in tissue engineering is the extrusion-based bioprinting, where we have a syringe that is filled with our bioink, one or two syringe, and through a controller is possible to construct the 3D scaffold layer by layer. As you can see, the typical workflow in order to produce this scaffold is obtain patient's image. Then from this patient's image, we are able to produce a CAD file. This CAD file is then transferred to other software in order to produce a G-code, and then the G-code file is transferred directly to the machine to be printed. As you can see here from the picture and the video that you can see. Here we can see different approach of a system where we can produce scaffold with well-defined topology. It's important to consider that we can have two different type of material that can be extruded. It's important to consider that we can have bio-ink or biomaterial ink. Bio-ink is principally composed by an hydrogel where the cells are embedded inside, so it's a bottom-up approach where the cells are able to fuse together and to promote their tissue regeneration. So it's the gel, the biomaterial is just like a shell for the cells that help them to promote their activities. Instead, the other typical approach is extrude directly the material, so the biomaterial ink, and then seed the cells on the scaffold. So the first is a bottom-up approach, the second is top-down. We focus principally on biomaterial ink. In order to select a biomaterial that can be used as ink for the development that can be used in extrusion-based bioprinting approach, it's important to consider, obviously, the biocompatibility, the cytocompatibility, the biodegradability. So all the physical and biological properties, but also are really strictly important to consider the rheological properties, such as viscosity and yield stress. Because if these are not well defined and well optimized, there is the problem that or the fiber, the extrusion is not allured, or when we extrude the materials, if it's too it present a low viscosity, we are not able to construct a structure that is freestanding. In this talk, I try to show you how we can use two different waste material. One is pectins derived from food waste, and the other is keratin that normally it's obtained by the waste of feeder chickens or feeder poultry. That is one of the most used meat in the world. And we show how combining and using this material, we are able to process, to transform this in biomaterial ink that can be used to develop a functional tissue. Pectin normally is, as I said before, obtained by waste, food waste, in particular from industrial citrus processing, is a biocompatible, biodegradable material, is not toxic, is eco-friendly, and is able to form an hydrogel, is soluble in green solvents such as water, and is low cost. And it's a really important waste in the food company and has a big cost for its elimination. The same is for keratin. Keratin is always, in this case, biocompatible, biodegradable. It's able to support cell adhesion and proliferation, is normally involved in intracellular signal pathways such as wound healing, apoptosis, has the capability to form gel, and also it's low cost. And it's important to consider that keratins, as reported in the report of some years ago of World Organization of Health, where they say that one of the principal causes of formation of ozone hole is due because the only way to destroy the keratin, the feeder chickens, is for incineration. So, because this material is biocompatible, find another way to reduce this waste that can be more green and can find more important application, can reduce this emission of CO2. Obviously, when we develop biomaterial inks from renewable materials, it's important to consider the risk of disease transmittance, in particular if we consider that some of this material can be obtained from animals. It's important to consider that can be some batch-to-batch difference. Normally, they have a poor technological properties for the extrusion and can be difficult to be processed. Startly, we start to analyze the keratin as biomaterial for tissue engineering and biofabrication approach. Normally, keratin we can find in different products from cosmetics, scaffold, medical products, or coating that already find application in different commercial products. But keratin can be used also in order to develop or skin in vitro models, in order that can be used to test the cosmetics, in order that can be used to validate their efficiency and reduce possible irritancy and toxicity of the cosmetic product, also to be developed leather cruelty-free in order that can be used in order to produce sweat or shoes. With a company close to Pisa, we optimize an extraction process based on alkaline hydrolysis, where we was able to produce keratin with a molecular weight measured than 10 kilodalton, and mixing this keratin with gelatin solution and cross-linked with glycidopropyl methoxysilane, that is a chemical cross-linker that is where after the evaporation of the solvent present in the solution is able to cross-link the keratin and gelatin, and then the scaffold can be rehydrated in cell culture media, we obtain a soft scaffold. So these materials, this combination of solution made of gelatin, keratin, and glycidopropyl methoxysilanes was, because our aim was to develop a skin model, was extruded using electrospinning process. Why electrospinning process? Because if we want to produce a skin structure, it's important to reproduce the typical micro and nano feature that compose the extracellular matrix of the skin. Moreover, it's a technique that is able to produce a large surface area to volume ratio. It's able to combine flexibility in the working process. It's a technique that has a large-scale production, and we can combine different materials at the same times, and is also an economic and versatile techniques. So how is it the approach? We have a classical syringe where between the metal needle tip and the collector, always made of metal, we apply a high voltage, so the application of this high voltage is able to allow the extrusion of the polymers the evaporation of the solvent present in the polymer solution, and in this case we are able to produce a non-woven structure. As you can see here, the application allows the creation of this combination of different nanofibers that, when are extruded on the collector, present this structure with nanofiber that comes from 2 nanometers until 10 micrometers. This was the machine that we have developed in our lab that was used in another European project, where we have an aluminum foil-like collector. There is the power supply for the application between the metal needle and the collector of high voltage tension. There is the syringe pump, and where that present the syringe filled with the polymer solution. So initially it's important to select the right solvent in order to have a well defined structure, in order that there is no trace of solvent in the final structure that can solve the single fiber. So we used the acetic acid. We optimized the ratio between gelatin and keratin, was in ratio 1:1 or 2:1, and also we try to optimize the different parameters that are involved in the electrospinning process, such as voltage, applied distance between the needle and the collector, the time of extrusion, and the flow of the syringe pump. As you can see from this table, all these parameters can alter the mechanical properties of the materials. So selecting the best parameters, we was able to obtain a structure that present an elastic modulus between 30 and 70 megapascal. That is an elastic modulus similar to skin. And as you can see here, this fiber that was obtained present a well-defined orientation, well-defined diameters in the range of 1.5 micrometers. On this structure, we seeded the cells in order to evaluate the real ability of this system to support cell growth. We seeded the human epithelial cells and also rat neuronal cells, because we want not only create a simple skin, but an innervated skin, in order to analyze better the influence of the compounds that can be tested on this structure, also on the nervous system that are present in the skin, to evaluate better the cytotoxicity, the toxicity of the compounds, and also the irritancy. And as you can see here, we have a good proliferation of human epithelial growth factor, and with the increasing time, a reduction of neuronal cells. This is due because when we fabricated this scaffold, we just created a scaffold that present the correct topological and mechanical properties for and biochemical stimulus for epithelial cells. We didn't print also neuronal growth factor that allow a better proliferation. But as you can see from this video, we was able to have, analyzing the confocal microscopy, that all the scaffold is completely filled with epithelial cells. The second material that we analyzed, as said before, was pectin. Pectin is normally derived from citrus peel and is normally produced like waste by food processing industry. As you can see, we can find pectin in the orange, mandarin, lemon, grapefruit, and As you can see, this is typical quantity of total of citrus production of citrus in the world, but there is a low use for other application. The particularity of this pectin is that they present a low methoxyl pectins that can be used for gelification because can added divalent cations such as calcium ions in order to induce the gelification. So now actually the pectin is used like targeted drug delivery system for colon pathology or for cancer treatment and gene therapy. Is also used in tissue engineering like microsphere for cell delivery or injectable gel for bone tissue engineering and wool dressing. But the problem with this material is that it presents poor mechanical properties that are not good for bioprinting technology. Normally they try to add other materials to pectin like pluronic acid or cellulose to improve. And we try to use another typical approach. Initially, we start to create scaffold, principally sponge scaffold, so a randomic structure, just mixing the pectin solved in water. Then we added the GPTMS. We heated it in order to create a perfect solution. Then we cast. We induce a freeze-dried process for 24 hours, and we obtain a porous scaffold. As you can see here, we try different concentration between ratio between GPTMS and pectin in the scaffold. And you can see here that changing the ratio between the pure pectin until increasing the quantity of cross-linker, the porosity try to decrease. And the range of this porous is between 50 and 150 micron. We analyze also the water uptake from the scaffold that we can see that increasing the quantity of GPTMS decrease because obviously decrease also the porosity of the scaffold, and instead the mechanical properties increase as we increase the quantity of cross-linkers, so of GPTMS. We tested, in collaboration with University of Pennsylvania, with Professor Gottardi, this scaffold with human mesenchymal stem cells, and we can see that biomaterial was biocompatible because all the scaffold was completely filled by the cells. So at this moment, we try to optimize the combination of pectin, the use of pectin in order to produce the biomaterial links. So what we did, we create our structure, we create our solution with pectin in adding GPTMS inside. We leave them to act for 4 hours, and then we analyze the viscosity and the printability of these materials, analyzing the yield stress. So we can see that normally when it is just pure, it's present at 0.5 kilopascal, and instead increasing the quantity of adding the GPTMS, this increased. What does it mean, this result? That we are able to produce a gel that when it's extruded can be preserved the extrusion form of the scaffold. So the other approach was we created our solution, we extruded the scaffold, we cooling at minus 20 Celsius degree, we induce a freeze-drying process. Here you can see the extrusion process at our scaffold. At the end of this process, as you can see here, we have not only the ability to produce micropores, but inside the line there is also nanopores. As you can see here, So we have a well-defined structure with micro and nano porosity inside the same. And this is a really good important because micro porosity is important in order to allow that all the cells that are growing on the scaffold are able to obtain to uptake all the nutrients that are present in the cell media culture. Instead, the nanoporosity is important to the cells to adhere to the scaffold and communicate each other. And as you can see here, using this approach, we was able to produce not only typical anatomical models such as ear or nose, but just joking, also the typical Piazza dei Miracoli. And you can see here that this material is stable also when we construct big structure. The other approach that we want, that we explored with this material is just using pectin structure like a template for fibrin gel. So we construct the pectin and then we covered with a fibrin gel where inside we embedded human bone marrow mesenchymal stem cells in order to study the formation of cartilage inside in this structure. So in this case, we analyzed two approaches. Initially, as said before, we produce our solution of pectin, then we added two different concentrations of GPTMS, and in particular in the first concentration of pectin, we also added nanohydroxyapatite. And as you can see, with this system of extrusion that we developed in our lab, we created a 3D scaffold with a well-defined topology. And here you can see the scaffold that we obtained as soon as that we finished to print after the freeze drying, and also after that we stored the samples. So there is a reduction with time. But when we rehydrated the scaffold, we can see that the topology and also the feature remains also. So we preserve the micro and nano porosity. And when we analyze with seeded cells, this is the fibrin just with the mesenchymal stem cells, human bone mesenchymal stem cells. And here at the different concentration, we can see that the metabolic activity increase a lot. So really the pectin helps the cells to activate their function. The other approach that we developed in collaboration with the Wasabi University of Technology was try to combine pectin with conductive fillers such as carbon nanotubes or PDOT or graphene. As you can see here, combining this material with different structure such as carbon nanotubes or graphene increasing the quantity, initially the film changed color, became more dark, because obviously we increased the concentration of the carbon nanotubes or the graphene. On these films, we try also to perform an electrical characterization in order to analyze what are the parameters of that influence principally the electrical properties. And as you can see here from this graph, increasing the quantity of carbon nanotubes or also the graphene, we are able to modulate the conductivity of the materials. On these materials, we see that scadiomyocytes, and as you can see here, in all this scaffold, we have a good adhesion and proliferation, but in particular, analyzing the scaffold with pectin, where we see that cadmium site with the multi-wall carbon nanotubes, there was a more defined organization and formation of cytoskeleton of these cadmium sites. So what are the future perspectives? For example, with this material, this was just a model of a vertebra with all the process such as vertebral body, fatty tissue, transverse process, or spinous process. And so all different combination this material with conductive materials or hydroxyapatite or just simple can be used in order to develop a patient-specific and multifunctional tissue. So, in order to create a really green biofabrication, it's important to start to consider all the waste product that the company that produce food or meat or other stuff that present a biological waste can be reused in order to develop a novel type of bioink or biomaterial inks that can be help in the development of a real functional tissue. Before to close this talk, I want to say thank you to you and to the organizer that invited me to give this talk. I want to remember that this year in Tuscany, it's organized by me and other my colleague, the International Conference on Biofabrication. So I hope to see you there. There is already the abstract, the opening of abstract. It's already done, so I hope that you can apply to this to submit your abstract, and we can see in Pisa and principally in Montecatini, because the Congress is in Montecatini. That is a real nice place in Tuscany. Thanks for your attention, and these are the principal sponsor of my research, and in the picture you can see all the staff that compose my group. Thanks again. Goodbye.
Portanto, acabamos de acompanhar o nosso último Keynote, agradecer ao professor pela excelente apresentação. A gente vai fazer um intervalo, daqui a pouquinho a gente volta com o final da nossa programação, será o encerramento da segunda edição do 3D BB, daqui a pouquinho a gente volta.